Okay, we're back. We're going to talk about each of these different types of organisms in more detail. And we'll start with the smallest one, viruses. There's some question about whether viruses are actually living organisms or not, because they cannot survive without their host and they're not capable of basic metabolic function. But viruses are found they infect all organisms. They can help transfer genetic material between organisms. They're very small, between 25 and 350 nanometers. And they're classified by the possession of either DNA or RNA and having either single or double-stranded nucleic acids. There are also prions, which are infectious proteins, and viroids, which are naked RNA. These can both be parasites of other organisms. And you can find viruses in groundwater, and a lot of viruses that can be found in the groundwater can actually cause human disease. So things like polio, hepatitis A, noroviruses, and enteroviruses can all be found in the groundwater. There are also rhinoviruses, which is a family of viruses that affect amphibians, fish, and reptiles. So moving on to the domain Archaea. Archaea are single-celled organisms. They have no nucleus, no organelles. They reproduce asexually by fission, and they are not known to form any form of spore. They are actually more closely related to us than they are to bacteria, which is shocking because they look a lot like bacteria. Many Archaea are extremophiles, but as we learn more about them, we're also finding them in a lot of common environments. And some of the most common types of Archaea are methanogens that create methane, sulfate reducers that reduce sulfate, halophiles, which love salty areas, and thermophiles, which love hot conditions. The bacteria, again, look a lot like Archaea, and they're also single-celled organisms with no nucleus or organelles. The bacteria are ubiquitous. There's a lot more of them. They are metabolically diverse, and many bacteria are pathogens that we find in water, some deep in the groundwater, and some used in bioremediation projects. The bacteria make up, along with the fungi, make up important microbiomes inside animals and plants, and they help organisms like ourselves with nutrition, metabolism, physiology, immunology, behavior, and fitness. And the best way to identify bacteria is using molecular methods, although we do have some morphological methods that we've used in the past. But only less than 1% of the bacterial species have been fully described. So working with bacteria is very challenging. Here's some common shapes of the bacteria that you might find in freshwater environments. So rods and spiral shapes, filaments, some colonial, some curved spheres, things like that. A lot of the bacteria that cause really devastating human diseases are found in freshwater environments, things like cholera, salmonella, E. coli, the bacteria that causes typhoid fever, which is Salmonella typhi, and Campylobacteriosis, which is caused by Campylobacter jejuni. So, but some of the bacteria are not necessarily pathogenic, and some are really important members of the group, the algae group. And the algae is this really broad term. Cyanobacteria used to be called blue-green algae. They are a photosynthetic bacteria, and they often reach bloom conditions, so harmful algal blooms sometimes in eutrophic water, so where there's a lot of nutrients. Cyanobacteria are some of the oldest known fossils, and a lot of them, I think almost all of them, can fix nitrogen. So that means they can take atmospheric nitrogen, nitrogen gas, and they can turn it into organically bound forms of nitrogen that then other organisms can use. They also absorb green light, which is interesting because most algae can't absorb green light. And when they bloom, they can produce toxins. So here's a picture of one of the earliest microfossils. This is what we think is a cyanobacteria, colonial kind of filamentous cyanobacteria, over 3.5 billion years old. In these fossils, they found 11 different microbial taxa. So they found Archaea, cyanobacteria, and then a species of gamma proteobacteria that they think consumes methane. These are fossils recovered from the apex chert in Australia, and carbon-13 values, which I will talk to you a lot about later. But basically, it's an isotopic method that helps us to determine that this is a bacterial origin. That's not just like some weird pattern in the rock, but that actually these cells were made by something living. So I'll talk about delta 13C values in other lectures. Here's some other pictures of the diversity of forms that cyanobacteria take. Nostoc is the kind of like leafy, lettucey looking shape at the bottom. Freshwater grapes are in the upper left. The first time I saw these was at Mount St. Helens, and I thought someone had dropped their blueberries in the stream. They were kind of purple where we were. You can see harmful algal bloom in a lake with just this really greenish sludge. A few more things about cyanobacteria. They have gas vesicles in them that allow them to float, and they can modify their buoyancy and also sink, which is really important because they have phycobilins, which are pigments that allow them to absorb this green light, and the green light penetrates deeper through the water column than other colors, and so it allows them to inhabit both surface and deep waters. They produce toxins and gelatinous coatings that make them not very tasty to other organisms. And the toxins that they produce can cause things like liver damage, dermatitis, and death in both humans and other organisms. They're also capable of photosynthesis at higher temperatures than other eukaryotic algae. So these are prokaryotic algae. A lot of the work of early algal ecologists was done by people who were working with microscopes, and this is Hilda Cantor-Lind. She was born in 1922 and is still working as a taxonomist, but also as an algal artist. So she has these amazing slides where she's placed these algae species in different orientations and taken pictures of them. The one on the left is actually Dinobryon, which is a colonial golden algae, and it forms that kind of colony by itself and actually can swim around as a colony to move the entire colony. So there's this really nice long history of taxonomists as artists as well. So the term algae is really a catch-all term. It's kind of like the term vegetable, like there's lots of different things you can throw into the vegetable category, just like the algae category. But for the most part, other than the cyanobacteria, they're non-vascular eukaryotic organisms, and they produce oxygen, so they're oxygenic, and they contain chlorophyll A. So the algae then provide a primary energy source for other higher trophic levels, for other invertebrates. But they can also be a nuisance. What you can see here on this kind of tree of life is just like all of the different places where you can find algae, and you find them up with the green plants, you can find them down, closer to animal type organisms, things like foraminifera and euglenozoa. So they're just really diverse, all of the organisms that we kind of lump together and call algae. So the first are some types of algae that are found in the plantae, with the plants. And so we have the chlorophyceae, the green algae, which is the most diverse group, and there's all different kinds of shapes and sizes. We have the charophyceae, or the stoneworts. These were the precursors to land plants, and they have a real rough feel to them because they precipitate calcium carbonate onto their cell walls. Red algae, the rhodophyceae, can live in deep water because they also, like cyanobacteria, use blue-green light. And then the glaucophyceae, or the microalgae, just really, really tiny species. And the chrysophyceae are the golden algae. They tend to live in oligotrophic lakes, and like I mentioned before, Dinobryon is one and can swim. Some other types of algae are kind of lumped in the chromoviolata. So things like Cercozoa, foraminifera, and radiolarians. Some of you might know these from marine ecosystems. The Dinophyceae, the dinoflagellates, which are free-swimming, mixotrophic, sometimes toxic. The Bacillariophyceae, or sorry, that's a hard one to say, Bacillariophyceae, the diatoms, they're dominant in oligo and mesotrophic lakes, and they can be both in the water column and on the benthos in what's called paraphytin, which I'll talk about in a minute. What's really cool about diatoms is that they produce a silica cell wall, and so they last for a really long time, and they're useful in paleolimnology and in forensics. And we can talk more about isotopes when we talk about how to date things. And then cryptomonads, which some are pathogenic. Here's just a few more pictures of the diatoms and their glass box-like frustules. Huge diversity, so beautiful, so many shapes and sizes. And again, because they're built out of glass, even after the organism dies, the frustule lives for it persists for a very long time in sediments. And then some algae are more animal-like. So things like phytoflagellates, dinoflagellates again, chrysophytes, things like euglenoids in the middle there, they can be both pigmented or colorless. They tend to prefer eutrophic conditions, and they have a red photosensitive spot, almost like an eye spot, but not quite. And numerous chloroplasts. And then the zooflagellates are things that are like parasitic protists, like Giardia, which is shown there, Leishmania, which causes leishmaniasis, which is like this kind of flesh-eating disease. And then things like amoeba, paramecium, vorticella, and Naegleria. Naegleria is a brain-eating protist that inhabits hot springs. So you got to be careful. Don't ever put your head under the water at a hot spring. And then finally, there are some selected protozoa. So things like the amoeba that I was talking about, vorticella is D, paramecium is F. So here's just some pictures of some of those protozoans if you've never seen pictures of them before. And a friend of mine, Basha Erland, is an emeritus professor at the University of New Mexico, and she does a lot of really cool art projects with rivers. So here you can see what she calls her water quality scrolls. So she would put an image of a microbe that causes disease on a scroll, and then she would float it down a river that's known for having that disease, kind of as like a conversation between the microbe and the water and the people that live in the area. So she has one for cholera, one for Giardia, and then a couple scrolls for E. coli. Algae, when it's growing on the surface of a rock, is called paraphytin, and it basically is this complex biofilm community made up of algae, bacteria, cyanobacteria, fungi, Archaea probably in there too. The algae can take lots of forms. They can be crustose, stalked, or filamentous. And the paraphytin helps to remove nutrients from the water column. It also supports river food webs, stabilizes the sediments, and modifies aquatic habitats. The word is a German word for paraphytin, Auffucht, which is an interesting term for kind of just like this biofilm community. Here's a picture showing what might be going on at the level of the surface of fine sediments or sands, different shapes of diatoms, different filamentous guys, different structures, different layers of this paraphytin community. Algae can also live in the water column, and when they do, we tend to call them phytoplankton. They're, again, microscopic, autotrophic, lots of diatoms, lots of different types of algae growing in the water column and collected in a totally different way. Okay, I'm going to pause and come back to fungi and plants in the next lecture.